Hello everyone, good afternoon. I'm Michael Mahmood. I'm the Mariculture Production Manager and Algologist at the Alutiiq Pride Marine Institute. I'm going to give you a brief overview on mariculture in the region. Alutiiq Pride is owned and operated by the Chugach Regional Resources Commission. It is a partnership consisting of 7 South Central Alaskan tribes located in Lower Cook Inlet and Prince William Sound. Here's a map of this region. The tribes are located in Cordova, Valdez, Chaniga, Alutiiq, Seward, Fort Graham, and Wallet. Chugach Regional Resources Commission, or CERC, is a native-led, community-based organization that is focused on sustainably managing our area's natural resources. Our executive director is Willow Hetrick Price, and she is in turn led by our board of directors, which is comprised of tribal leaders from each of our 7 tribes in the Chugach. Here's a photo of our main hatchery and the happiest place in Alaska. Here's a photo of part of the hatchery. This is one of our main areas. There's some large algae culture tanks and some downwelling units where most of our clam mariculture takes place. Each year brings something different to the hatchery. Our main focus the past couple of years has been culturing butter clams, little neck clams, and softshell clams. More recently, we've been working with geoducks, razor clams, sea cucumbers, abalone, and king crab. In 2020, we also became a kelp nursery. We have led the way in our region with help from Green Wave, a nonprofit based in Connecticut, to provide kelp seed string to anyone wanting to become a kelp farmer. Farmers will collect sorus, it's the reproductive material, and send it into APMI. We then induce spore inoculation, which in turn grows on the string and develops into kelp in this picture. Those are all individual kelp blades. We mainly grow sugar, ribbon, and bull kelp. Once it's ready to be outplanted, we sell these spools back to the farmer, and they outplant in the ocean, and it's then outplanted around now in October, and it's grown throughout the winter months and harvested around April or early May. This is a broad overview of our shellfish work. Each spring, we collect broodstock from a particular area. These get brought back to the hatchery and are spawned. We then grow them through their larval phase, where they are continuously fed microalgae, which also grow at APMI. And we grow them until they metamorphose into juvenile clams. We'll continue to feed them until they are large enough to outplant at the beach where the parent broodstock came from. Come springtime the following year, we might return to the beach to see how survival went. Depending upon the experiment, we plant shellfish in small PVC tubes, in wooden mesh covered boxes, or if we seed a large area, we can cover the shellfish with a fine mesh net to prevent predation. We also do clam surveys. This is an example of a habitat suitability survey. The beach is plotted out into grids, and each grid is then surveyed for the population of existing clams. From this, we can extrapolate if the terrain is suitable for survival, and if an area is also able to welcome an enhancement of freshly grown hatchery clams. We can also mark our clams at APMI with calcine. Calcine is a non-toxic, permanent dye that is absorbed by the growing shell as calcium carbonate is deposited. This mark can be seen by using a fluorescent light microscope. We've also successfully used this to mark sea cucumbers, and this is a useful tool in differentiating wild grown from hatchery raised when surveying populations. This is how I get my 15 minutes. One of our niche species at APMI are bidarkies, or black katydchittens. It is a subsistence food item in our region. We still have a lot to learn, but we have managed to successfully spawn a few and managed to get them to the juvenile stage, but it's going downhill from there. So we have a lot of work to do. They're pretty cute. APMI has the ability to host and work in partnership with other scientific institutions to do a variety of research. These include the Sea Life Center, University of Alaska Fairbanks and Anchorage, and Alaska Pacific University. This past summer, we worked alongside Dr. Jessica Glass from UAF in an eDNA project which examined RNA from butter and littleneck clams during their larval stages. Currently, we have an ongoing study led by the Canadian Department of Fisheries and Oceans. And of all the species I get to work with, sea cucumbers are by far my favorite. There's a lot to be discovered. We don't know how old they are, or how old they get to be, or when they become sexually mature. So this is going to help in that project by extracting DNA. Last year, we worked alongside UAA and participated in a study to observe ingestion rates of microplastics by blue mussels. And later this year, we are going to begin a project which will look at the ingestion rate of soft-shell clams of hydrocarbons from different types of fuel. In the past, in partnership with NOAA, APMI has done extensive husbandry involving red king crab. We've collected female crabs with fertilized egg clutches. These eggs are then hatched in Seward, and we've been able to raise them through their 4 zoeal stages, where they then metamorphose into juvenile king crab. Last week, we went and collected golden king crab, and we're going to try and replicate this study with that version of the species. So these are some eyed eggs, and then those are newly hatched larvae, and then that's about 2 months later, it's a little baby king crab. Finally, we have our ocean monitoring program. This program relies on citizen scientists in our tribal communities to monitor the health of our oceans by examining ocean chemistry, monitoring harmful algal blooms, and shellfish biotoxin levels. Our next presenter, Allison Carl, will expand on these topics. Finally, to conclude, I wanted to highlight that last month, CERC purchased the building and land that APMI sits on from the city of Seward, and we couldn't have done this without help from our partners at the NDN Collective and the First Nations Development Institute. That's my presentation. Are there any questions?
So,
anyone else?
so the king crab, you raise them up to a certain point and then sell them to what?
So back in the past, in the olden days when I first started working back in 2012, this project was just ending, and it was Fish and Game would not allow us to release them back in the ocean. So mainly they went to different institutions to do different experiments.
Oh, okay. So it was not a commercial?
It was not.
Yeah.
But they wanted to learn what if there was a giant die-off,
yeah,
and we needed to start helping the population along.
Okay.
So they were not able to successfully breed in captivity, so we would just collect clutches, females with already fertilized eggs.
Didn't AJ Paul do a bunch of research on that as well?
Mm-hmm. Yep.
But,
And he was before my time.
yeah, I wouldn't understand that. But. same thing, they wouldn't let them go into the wild?
Yes. So around 2018 or 19, the Fish and Game finally said, okay, we'll let you release them. But they had to be, divers would dive the juvenile crab down there, only this big. And they were super glued with some fishing line, kind of like around a maypole. Because the Fish and Game didn't want them to spread out. And then the next year they said, okay, that was a dumb idea. We'll let you release them. But then a government shutdown happened.
Oh,
And then nothing happened.
well, they're looking for dumb ideas, I got it.
Okay.
Is it the same with the golden king crab?
So the golden king crab, this is just, we got permits to collect them. And the red king crab, when they go through their larval phases, we have to keep them fed. But the golden king crab have a yolk sac attached on their larva. So I don't have to worry about making food for them. So it might be a little bit easier for survival.
But you still won't be able to release them,
No, this is just a,
just,
take a look-see.
right.
And why wouldn't they want you to let them go?
Things raised in
Yeah.
cap, same thing for Sea Life Center, like things that are raised in a captive environment, they don't want to take a risk of introducing something from this facility into the, or from a facility.
Yeah, I mean,
At first.
Cooties.
Ocean cooties.
Mike, how do you get them to spawn? Do you inject hormones in the
The
water?
what?
I don't know, different species,
Oh.
like,
Dirty.
to trigger spawn.
Yeah, very, very white. No, for clams, we usually warm the water and throw a bunch of algae that we grow in.
Okay.
And that usually does the trick. Sometimes we can use serotonin, but that's a certain case. But usually it's just warm water and food.
Do you guys do oysters like that?
We do oysters we do oysters just to keep our permits valid, but a lot of the oyster farmers in Alaska get their seed from Homer, from KMSA, or from Southeast, and they do a fine job of distributing. But if there was a farmer that started out here in Res Bay, we could supply them.
And so do you do much commercial revenue stuff, like
Just
the kelp or?
Yeah, mainly kelp, and that's still very new.
Is there a kelp farm here in Resurrection Bay now?
There was, not anymore, there was a farm out by Pony Cove for a winter, but it was too far for it to last.
Sea cucumbers. Fascinating that you're fascinated by sea
Yeah,
cucumbers.
they're cool.
Well, they're not trainable. Are you doing it for commercial purposes? Are they involved in subsistence? I know people eat
So
them
there's a dive
themselves.
fishery in Southeast, and in the past we've worked with them to try and enhance the population, but they couldn't really come to an agreement with what they wanted to do. So a lot of it was just student research in the past, work with cucumbers, and then every so often we'll get a phone call from someone who wants to become a sea cucumber farmer, and they have dollar signs in their eyes. So it's just something to work with to, they're just their larval phases are very interesting to me.
Are there commercial operations?
There is
Diving
not,
is
there's just a
grabbing.
dive fishery at this point.
Golden king crab are a deeper species than red king crab, and so do you guys have methods to try to work with that? Or?
We are chilling our water, and we have them covered. And they've only been in the hatchery for a week, and they haven't died yet.
They seem happy.
Yeah,
But you're working on it.
I I get hungry.
I'm just curious what the microplastic study and what that was aimed to do.
You know, that was something that I was not involved in. I'm not quite sure what the outcome of that was, but I know that paper has not been published yet. So be on the lookout.
They weren't there for very long.
No, it was like a week, 2-week study. But I do know we got some very finely ground microplastics. And then added them to the tanks where the blue mussels were. But I'm not sure what happened there.
Maybe this was probably like, maybe 10 years ago, but someone was saying that shellfish were having trouble building their shells because of difference in temperatures. Do you know, are you also involved in monitoring for environmental impacts? Yeah.
I believe Allison is going to talk a little bit about ocean acidification, but we do have a machine at the hatchery called a percolator, which is doing continuous monitoring of calcium carbonate, pH and temperature, and there's 10 of those throughout the state. Seward has 2 of them.
World.
Wow.
Yeah.
What?
There's 10 in the world, there's 4 in
Oh,
the
10 in
state.
the world.
Sears or Chem Lab.
Oh, wow. Yeah.
And processes all the OA samples.
But when that was hitting like Whiskey Creek and the oyster hatcheries in Oregon and Washington State, we really got concerned that Alaska was next. This was like 2013, 15.
Yeah.
And then so we were starting to dose our water with soda ash, but then it never really materialized up here. And it never, we got lucky. And I think it's gotten a little bit better down in the lower 48. But it was like kind of, like it was right around the same time as when that log
No, I wonder if that was back.
and all those mirrors died.